Hi, I'm Shama.
I'm Orla.
And we've been working at the Neuroscience Department at UCL for 2 weeks.
Here's a compilation of video footage from our time at UCL.
The lab we were exposed to investigated a distinguished form of behavioural neuroscience, specifically the development of neurocircuits in the hippocampus and how they formulate a critical part of memory and spatial navigation. The research primarily comprised of investigation of in vivo electrophysiological recordings of the neurons and anatomical tracing. We constructed tetrodes which are implanted into a specific area of the brain to formulate traces which can distinguish place cells, head cells and grid cells from others. We were given an atlas of a rat brain and taught about the cuts to the tissue, such as sagittal and coronal, and then used a cryostat to slice the coronal sections of the brain and to locate the AD and DVN, two entities we have been instructed to find. The entire histological process was extremely time-consuming and consisted of the mounting of the brain slices onto slides, staining, and adding a glass cover slip.
Hi, my name is Joshua Bassett, and I do behavioral neuroscience research here at UCL, and my field is studying how the brain represents space. And what I mean by that is if you walk into a classroom or a restaurant or something for the first time, it won't be very long before I could come up and blindfold you and ask you questions like, where is the exit? How far could you walk before you bump into a wall? And you'd be able to answer those questions with very little exposure to a new environment. And if you think about it, that's kind of a complex thing that your brain has just done there, because any spatial environment has a lot to it in terms of the basic sensory information that we can take in. And the reason we're interested in this is that spatial representation in the brain is related to a lot of things more generally. It's related to how we make memories in general about our lives and learning and all of these kind of things. And so the hope is that we can help not only people have specifically spatial problems, but in general memory impairment and just to understand broadly how the brain does what the brain does. I really liked having in2science students in the lab because they haven't heard all of my stories already. To be serious, those of us who work in science are really enthusiastic about it, and it's nice to be reminded what it's like when it's new to you, and to sort of remember that enthusiasm. And the in2science folks they don't have to be here, they're here because they want to be here. And so it's pretty fun to be able to share your enthusiasm for the subject with people who are here because they're interested in it too.
Lastly, we'd like to say thank you to the in2science team for giving us such an amazing opportunity. It was truly an experience that we would never forget. Thank you so much.